Good afternoon, everyone, or good morning if you're further west. I'm Jane Wall, Director of Business Development and Membership Services here at BIA. Welcome to our BIA member webinar on the scale-up of continuous chromatography, led by Dr. Peter Levinson of Pall Life Sciences and Dr. Mark Brower of Merck. Continuous bioprocessing for biologics manufacturing is being adopted in the biopharmaceutical industry by big players such as Merck. Pall Life Sciences has partnered with Merck to help bring this innovative technology platform to fruition. So we're fortunate to have these two key players here today to tell us more. Just a couple of things I need to mention before we start. For your information, everyone is muted to reduce background noise. We'll take questions and answers at the end. Please use the question box in your control panel and state who the question is for. This webinar will also be recorded and uploaded onto our YouTube channel, and that should be up there tomorrow for you to send on to colleagues. So I'm going to hand straight over to Peter Levinson from Pall Life Sciences now. Over to you, Peter.
Thank you very much, Jane. So good afternoon to everybody, and I also echo to those who are west of us, good morning. So I'll give you a brief overview and an update on how we see continuous bioprocessing and how it has evolved over recent years, and then allow Mark to give a much more in-depth presentation on scale-up of continuous multi-column chromatography. So I'll talk about some of the core technologies that we have developed and worked on to enable continuous bioprocessing, how unit operations can be integrated to go from batch to continuous, and then talk about the scale-up of the various platforms that are available to us. Okay, sorry about that, the slides weren't moving. So there's been widespread acceptance and use of single-use technologies over the past 20 years or so. And this has really underpinned the movement to move from batch downstream processing through to continuous. And if you look at this slide, you can see that we've evolved from single-use filtration, single-use chromatography, through to various biocontainers, sterile connectors, bioreactors, multi-column chromatography, clarification. All of these established single-use technology which can be leveraged to facilitate continuous manufacturing. And continuous processing has been used in many industries for quite a number of years. The glass industry, petrochemicals, automotives. Many of these industries have moved from traditional batch processes to continuous with this concept of one-piece flow. And building of a car is an example, where you start with the various components and you gradually move through the production line, and a car drives off every minute or two. And this has been really pioneered very much by Toyota and the concept of lean manufacturing and reduction of waste. And there are a large number of wastes in manufacturing. We see them in pharmaceuticals as well as other industries. And if you can eliminate the waste, you can operate in a leaner mode, and that has been demonstrated to give higher quality and productivity, typically within a smaller footprint. And this is how we see continuous processing. And now if we look at the current batch process, and in the top panel here, we've got cell culture, clarification, typically centrifugation, followed by depth filtration, a series of capture and polishing chromatography steps, and then the UFDF stages, a fairly conventional method of purifying a recombinant biological molecule. And if we go to a continuous process, we're using the same basic unit operations. We have cell culture, we have clarification, we still do the chromatography, but we'll now do it in a continuous mode, and we can do the UFDF in a continuous mode. And the beauty of continuous processing is that we still use the same filters, the same buffers, the same chromatography media that are used in batch. All you're doing is doing the process in a slightly different way. And I would refer to it as an evolution rather than a revolution. And there are concerns. There are concerns from the regulators. Is this a step too far? This slide is one given by the FDA in August last year. And you can see that they are supportive of continuous manufacturing as much as they can be. Janssen Pharmaceuticals, there was a process licensed last year, which was a batch process that has now moved to continuous. So the regulatory authorities are warming to the technology and the processes, and they are now attending and presenting at various conferences and events around the world. So our journey to move towards continuous processing has really been underpinned by three core technologies. A technology based on acoustic wave separation that we have licensed from a company called Flow Design Sonics that enables us to do continuous clarification of cell culture. Multi-column chromatography using the BioSMB technology that we acquired from a company called Tarpon Biosystems, and that has a multi-column valve cassette that is single use. Mark will describe it in much more detail soon. And then the final area is continuous filtration, where you can use single pass tangential flow filtration to do both UF and DF steps. And again, we have exclusive licenses for that, practicing that technology from a company called SPF Innovations. So these three core technologies can be used to enable you to do continuous processing. And in order to demonstrate it can be done, it's very easy to talk about it, but the real proof is in seeing it. So we constructed a laboratory in our Westborough, Massachusetts facility. This is a photograph of it. This lab contains all the components and equipment to go from cell culture through clarification, through the capture chromatography, virus inactivation, through polishing, through concentration, diafiltration, and final sterilizing grade filtration. The dimensions of this lab are 6 meters by 6 meters, so it's a 36 square meter laboratory. And about a year ago, we were pleased that we could make just over 2 grams an hour of pure monoclonal in there, so around 50 grams a day. Well, since then, we've intensified it. We're now able to produce in excess of 100 grams of monoclonal in one day in this small laboratory. And 100 grams is quite a significant number because you're now moving into the realms of the amounts of antibody that you may need for early stage clinical production, all within this very small footprint. And we've presented this data at various conferences, but just to give you one snapshot of it, this was the trends of the critical quality attributes when we did produce the 100 grams of antibody. So you can see in the top panel the host cell protein, big reductions with the protein A step, and then more reductions with the anion exchange polishing step. Aggregate levels were reduced down to acceptable levels, and we were able to produce in excess of 100 grams of antibody in just under 21 hours. So it can be done in this very, very small footprint. So the question that we then faced was, how far can continuous go? It's one thing doing things on a laboratory bench, but in order for it to be continuous bioprocessing to be fully adopted and accepted within the industry, then we need to be developing and introducing large-scale GMP compliant systems. And we have a lot of experience in scaling up filtration technologies and membrane chromatography, but how do you scale up the acoustic separator for clarification, and how do you scale up the multi-column chromatography system for purification and polishing? So just to give you a flavor of it, this is data which we've generated on scaling up the clarification. So the benchtop system operates at 4 liters an hour. We made a prototype chamber which clarifies at 11 liters an hour, and the chamber that we're going to go into production with operates at around 50 liters an hour. And these can be manifolded together both in series and parallel to give you much higher flow rates to enable you to process the contents of a 2,000 liter single-use bioreactor within 6 to 8 hours, something of that order. And then scaling up the multi-column chromatography, and this will be the subject that Mark will discuss in much more detail. But so this is the BioSMB process system. It is the big brother of the BioSMB PD system, and this is a system which will operate under GMP conditions and will operate at up to 350 liters an hour. And that we introduced in October last year. We're also in the very final stages of development of a continuous virus inactivation skid. I say continuous, it's semi-continuous. We have two mixing tanks, one which is filling while the other one is undergoing an acid hold. But with this system, we will be able to hook it onto the end of the capture chromatography step, and then the output of it going on to the polishing steps. And this system we will have on show at the BIA conference in Boston in the end of September this year. And then the final area that I just wanted to share with you is really what our pipeline of products is like, and the sort of commitment that we've shown already and where we plan to go. So we have the benchtop Cadence acoustic separator available today. The full-scale GMP system will be launched before the end of this year. We have the BioSMB PD and process systems already available. The virus inactivation skid will be available again towards the end of September. We have a new range of virus filters that we introduced last year, which perform very well in continuous mode. We're developing an acoustic wave separation system for clarification of fruition culture, and the benchtop system will be available later this year. We have single pass TFF and a product called the inline concentrator, which will do concentration continuously, and we have developed an inline diafiltration filter, and that we've done extensive beta testing with over the last 12 months or so, and that will be commercially available again later this year. And we still have more to go. We will still be introducing other technologies, other products for fusion applications, as well as working hard on process monitoring and control, automation and validation. I'm going to hand over to Mark and let him speak in detail about the scale-up of the BioSMB process. Thank you.
Well, thank you very much, Peter, for giving us a great overview of the continuous technologies currently being developed at Merck, at Pall. For the rest of the webinar today, I would like to talk a bit about the scale-up activities that have been going on at Merck with the continuous chromatography system, the BioSMB. And as you can see, the name of my talk is Scale-Up of Continuous Chromatography using Cadence BioSMB Process System. But really, the talk covers the scale-up aspect of going from PD to process. So I wanted to start off the talk today by not taking for granted that everybody in the audience knows about continuous chromatography systems and how they work.
Is it not changing?
It's not changing.
Is your internet connection okay?
Yeah, I'm on a corporate network.
Yeah, it's not moving for me either. Yeah, that took a very long time, didn't it?
I mean, is this just a...
I'm going to hand over to Mark and let him speak in detail about the scale-up of the BioSMB process. Thank you.
Hello, and thank you very much, Peter, for talking about the continuous unit operations that are being developed at Pall. Today, I'm going to focus mostly on the scale-up of a separation process on the BioSMB process system and compare that to the process scale BioSMB system. So I wanted to start off the presentation today by talking a little bit about the background of continuous chromatography and make sure that we're all talking the same language as I go through the rest of the presentation. There are several multi-column chromatography systems on the market today. I will be focusing on the BioSMB, and the process scale system is shown in the upper left-hand panel. Right below there, you can see that the BioSMB can accommodate multiple columns on the valve block cassette. The purpose of that cassette is to direct flows to the different columns throughout a separation process. And the concept here is really to maximize the loading on the columns while also doing the individual steps of a chromatographic separation on other columns at the same time. So as you can see in the box, there is a load zone that can consist of multiple columns, in this case 4, where the feed is distributed over one or more columns in the first pass, and that feed is then sent over to columns in the second pass, which again can contain one or more columns. In the picture shown here, it's a 2x2 configuration. This allows you to capture any material that flows through the first column onto the second and has the functional characteristic of getting higher dynamic binding capacities on those columns compared to a batch configuration. We tend to visualize the method itself in terms of a circular chronograph. That's shown in the upper right-hand panel, where the inlets to the column are shown in the outer donut of the circular chronogram, and the effluent is on the inner donut. And then the columns are represented by the spokes. So in this instance, if you look at column 4, you can see that it is receiving fresh feed, and the effluent of that column is going to the second pass. In this instance, column 2 would be receiving that second pass, and the effluent of that column would be going to depleted feed as flow through. And therefore, you can see that at different periods of time, columns 5, 6, 7 would be going through wash, elute, strip, and re-equilibration steps. So a little bit about the historical perspective of scale-up at Merck. We started with baseline processes using bottles of feed out of the freezer on the process scale system in the laboratory. Here we were using 1 or 5 mil columns and operating at relatively low flow rates. Once we became comfortable with the methods, we scaled up to harvest 100 to 200 liter fed batch bioreactors and continuous downstream processing. And in this instance, we were only required to scale up to 2.5 centimeter columns with approximately 40 milliliter column volumes. Still not large scale compared to current industrial processes. After that time, we started focusing on perfusion upstream processes. This actually required us to scale down somewhat to approximately 10 milliliter columns to balance flow rates coming off of that perfusion bioreactor. But what we really need to do to advance continuous chromatography to the next level is show the application to GMP scenarios to harvest 2,000 liter bioreactors. So again, the purpose of this presentation is to focus on the 400 liter harvest of a batch bioreactor taken through a step in a reasonable 12-hour time frame to come up with appreciable amount of product. So the focus of this experiment was really to test multi-column chromatography at the process scale and to compare that process with that being run at the process development scale with the same feed at the same time. So definitely a comparison between the scales and with a batch reference process. Here's a picture of the BioSMB process system. It is constituted of 4 different modules. 2 are the electronics. One is a power module. The second is the control module. And then the 2 in the front, which you can see in the picture, are the fluid management or fluid delivery on the left. And that consists of 7 pump heads and pressure sensors, as well as bubble sensors. And then on the right, you have the fluid distribution module, and that's where the cassette is housed. And again, the cassette is a traffic cop and directs fluid to the correct column at the correct time. So this skid was developed to service 2,000 liter bioreactors and can accommodate flow rates up to 350 liters per hour processing, really designed to get you to be able to process your batch within a shift. It can accommodate up to 8 columns with 7 different buffers or process solutions and 5 outlets, very similar to the capabilities of the process unit, with each of those outlets containing UV, pH, and conductivity sensors. If you look at the valve block itself between the pilot scale and the process scale, the pilot on the left contains many valves that are shown in the ovals, the orange ovals there. And again, those open and close to direct the flows to the correct column during the process. In the instance of the PD scale unit, the valve block can accommodate up to 16 columns and operate at 12 liters per hour. The same basic valve technology has been applied to the process scale. However, it can only accommodate up to 8 columns and has a significantly higher flow rate of 350 liters per hour. One of the main differences, however, is loading the valve block. As you can see in the picture on the left, there are multiple screws and washers that need to be replaced to change out the cassette in between campaigns. However, the unit on the right is installed by pneumatics, and the entire single-use manifold that's used in the Cadence BioSMB process scale system can be replaced in 30 minutes as a design requirement. So what I'd like to move into is process characterization and how you design a continuous chromatography step. We've done it in 2 ways. The first is a very classic method using batch breakthrough curves. So this is a technique that's used in many process development labs today. In this instance, we did breakthroughs at 3 different linear velocities, got the breakthrough curve, and back calculated the Qmax and the mass transfer coefficient, KOLA. And you can see the values on the right-hand side of the screen obtained from this analysis. A second method for coming up with these parameters is to run experiments on the BioSMB and the multi-column configuration for the capture of the protein in an actual continuous chromatography method. This method requires much more material. However, when you look at the load capacity and versus the capture efficiency from these experiments at different flow rates, you can also back calculate the Qmax and the mass transfer coefficient very nicely from this analysis. And here you can see less than 2% difference in the two methods between the Qmax that was observed, the maximum static binding capacity, and the mass transfer coefficient KOLA. These two values can then be fed into equations to help predict where you would operate the continuous chromatography method. So what's plotted here is an operational curve derived from those two parameters, the KOLA and QM, and it's telling you basically the required contact time or residence time in the column for a given loading. So you can operate anywhere on this line and achieve a 99% capture of your protein. We chose to operate just above 46 milligrams per milliliter loading, giving us a residence time of about 3 minutes. Shown here is the method editor and how methods are created for the BioSMB, both at the PD scale and the process scale. I'm happy to say that some of the rounding errors that are present in the current PD scale software are, have been corrected in the process scale software, but the basic interface is the same between the two systems. So here you can see you put in each of the different steps in the chromatographic separation, such as feed, wash, elution, CIP. You put in the corresponding column volumes and flow rates, and the flow rates can be defined as either liters per hour, milliliters per minute, or centimeters per hour. And then define the outlets of where those streams are going for very easy method generation. And this is just basically a comparison between the two scales of operation that we ran our experiments. So at the PD scale, we operated at 4 mils per minute, whereas at the process scale, 37 liters per hour. We moved from 5 by 5 mil columns at the PD scale to 5 by 770 milliliter columns for a total of 3.9 liters of protein A resin. Here again, we operated both of these scales at a residence time of about 3 minutes with a 5-column method and an operating binding capacity of just over 46 grams per liter. And just to highlight, this was 150 times scale up, and we were able to process 400 liters in 13 cycles over 11 hours. And I mean, the real accomplishment was that all of this work had been done in a 3-week time period, from receiving the small scale columns to processing the 400 liters through the system. So now a little bit on the results. What I'm showing here is cycle to cycle consistency. On the left-hand side, you have the overlay of 13 cycles of operation on the process scale system, showing the UV elution peaks. Each of those peaks represents the 13 elutions from each of the different 5 columns that were attached to the system in this method. On the right, on the left-hand panels, you actually see HCP, DNA, and aggregate levels in the effluent pools and the product pools for both the process scale in blue and the PD scale in red. So again, the PD scale was only run for 8 cycles, whereas the process was run out to 13 cycles. So you can see very comparable HCP profiles coming off of these columns between the PD and the process scale. And the same can be said for DNA. We do see one outlier in the data, that's cycle 6 on the process scale, that's showing pretty high DNA levels. We're still trying to investigate if that's a real measurement or an outlier in the assay. The aggregates for both of these processes resulted in less than 1% and very comparable between the process and PD scales. And a little bit on the column-to-column performance. So on one of these cycles, the column effluents were fractionated and tested. So this table shows the columns as tested prior to running, so the number of theoretical plates shown in the leftmost column, as well as the asymmetry and the delta pressure in that column. However, if you look at from the process side, the elution pools were very consistent at 3.3 column volumes, and the concentration of the antibody that was captured here was, within 10%, 13 grams per liter in those elution pools for each of the individual columns. If you were then to look at the HCP, DNA, and aggregates for those elution pools, again, they're very similar and basically indistinguishable from the data that was shown on the previous slide. This is basically a summary, again, and showing the scale up from the PD to the process system, the Cadence BioSMB technology, in reference to the batch process. So here we were able to increase the product concentration by overloading the columns from 9.5 grams per liter up to 13.8 grams per liter, and very consistent between the two scales. We also had very consistent yields at 97%. And again, as discussed previously, very indistinguishable aggregate DNA clearance and HCP clearance. Going from the batch process that had a specific productivity of 16 grams per liter hour, we were able to increase that approximately 3.5 times to 56 grams per liter hour with the both the Cadence BioSMB PD and process scale processes. And again, on the bottom, you can see the process parameters that were used for each of those scenarios. So in conclusion, I'd just like to say that we were able to purify 2.3 kilograms of monoclonal antibody from 400 liters of broth in 11 hours and 13 process cycles. We only used 3.85 liters of protein A media for this run, and this is actually only approximately 10% of the maximum capacity of the Cadence BioSMB process system. All of this work that was presented here was conducted in 3 weeks, so I think one of the messages we're trying to send is that continuous process development doesn't have to be a lengthy process as long as you have a methodology behind it. And the end result is a very scalable process from the PD bench to the process scale. And with that, I'd like to open up the floor to any questions, but only after flashing the acknowledgements. So this work wouldn't have been possible without significant contributions from the Merck team, specifically Nuno Pinto, Doug Richardson, Bumit Patel, and Jun Hio, as well as Sen Zhu, Mike Kazola, and Egg Lawacki. The Pall team was also instrumental in pulling this off and logistics around getting the BioSMB to our labs. Their team consisted of Mark Bishops, Carl Rogler, Fabrizio Palella, and Alexander Klawich.
More questions at the moment. I'm sure that if you had any more questions for the presenters after this, we could pass them on, but I think I'll close that section now. Apologies for some of the connectivity issues in the second half there, the challenges of transatlantic webinars. But thank you to both our presenters, really interesting presentations. just a couple of slides from us. If you're not already a member of the BIA, please contact me for further details. We advocate on behalf of the biotechnology sector in the UK, and our members also benefit from discounts to key UK and overseas events and significant savings on laboratory equipment. Also significantly, in terms of manufacturing, one for your diary, the 14th Bioprocess UK conference will be held in Cardiff this year on the 29th to the 30th of November. Make sure it's in your diary. This year the title is Bioprocessing, Delivering Innovation to Patients, and the PAC programme includes a dedicated workshop on purification of next generation medicines. Please see our website for further details, or let us know if you'd like us to send them to you. So that's it from us today. Thank you very much for tuning in.